Hello everyone. I'm your host for today's webinar, Mike Lasecki. Today our topic is capturing movies of molecules using ultrafast X-rays. And we're going to hear a lot more about Arizona State University's compact X-ray laser project. This webinar will be recorded, and yes, you'll automatically receive a link to the recording. It usually takes us a couple of days to get that out to you. You'll be in listen-only mode, so please use that upper right-hand chat window to ask questions of our expert panelists. Let me tell you that this webinar is hosted by ATE Central. ATE Central acts as an information hub for the National Science Foundation's ATE grantee community. You can learn more about ATE Central and all of the webinars they host at the URL shown at the bottom of the screen. This webinar is brought to you by the Nanotechnology Collaborative Infrastructure Southwest, NCI Southwest at Arizona State University. They're the regional hub of the National Nanotechnology Collaborative Infrastructure. And also bringing us this webinar today is the NAC Network at Penn State University. Thank you to both of those organizations for bringing us this webinar. As I mentioned, I'm your host, Mike Lasecki. I work at Luca Partners, and I work with both ASU and Penn State in producing these webinar series. Joining us today is Trevor Thornton. He's a professor at ASU and director of the NCI Southwest. Trevor, would you say hello to everyone?
Yes, thank you, Mike. Hello, everybody. We're here in the beautiful new building that's been built for this compact free electron laser that Bill will be talking about in a few minutes. It's a beautiful day, and I'm very excited and happy that Bill was able to take time out and explain the building of this exciting new facility here at ASU.
Good, thanks, Trevor. Bill gets his own building for his laser. That's pretty cool. So also joining us today is William Graves. He's an associate professor at Arizona State, and he has a dual affiliation, first with the Biodesign Center for Applied Structural Discovery, and he's also a professor in the physics department, so great background there. Bill, you joined ASU, I think it was 2015, and you came from Tech out on the East Coast, huh?
Yes, that's right. Thanks for having me today, Mike. Very happy to be here.
We're really looking forward to the presentation, Bill. Everyone, I'm going to advance to Professor Graves' first slide. There we go. And Dr. Graves, go ahead and take us forward into the webinar.
Okay, thank you, Mike. Okay, yeah, I'm going to tell you today about this new X-ray source that we're developing, and our goals here are to bring the power of the large accelerator-based X-ray sources into laboratories such as a university like ASU, or into medical institutions or industrial labs. So, as you'll see as I explain over the next few slides, X-rays started in small labs and then migrated to the national laboratories because of the scale of the facilities, and we hope to migrate them back out again. And so I'll give you some background about what we use X-rays for, and that sets the stage for where we're headed with them. And so historically, X-rays have always been used for one of two things. One is to look at things we can't otherwise see, and what you're seeing on the screen now is a Nature paper from 1896, was the very first publication of an X-ray image, and that's Frau Röntgen's hand there with her wedding ring on her hand. And Wilhelm Röntgen was awarded the very first Nobel Prize for the discovery of X-rays. That image is interesting because it shows us both what you can do with X-rays and what before now you haven't been able to do. And so we see inside, we see below the skin and the flesh, and see the bone in our hand, but we don't see many things. We don't see any tendons, we don't see muscle, we don't see blood vessels. There's lots of soft tissue that's not visible. And the reason for that is it's all about the same density. The bone with calcium in it is a heavy material, heavy atomic weight, and so it stops more X-rays, and so we see it as a shadow. And so what this image is a shadow image of the heavier elements, the gold in the ring and the calcium in the bones. And one of the things that we're moving toward is the ability to image soft tissues. So X-rays are used to make visible what's invisible otherwise. And then the other thing that they've historically been used for, using these Nobel Prizes as examples, this award was made to father and son team, Lawrence and William Bragg, in 1915, won the Nobel Prize in Physics, essentially for the discovery of atomic structure of materials. And what they were able to do was diffract X-rays off of the atom lattice in NaCl, which is normal table salt, and tell the positions of the atoms in the salt and discover its structure. And so X-rays, because the wavelength is very short, it's just another form of light, but the wavelength is about an angstrom or so, which is about the size of an atom. And so it lets us see down to the atomic scale. And so those are the two things that X-rays have really been used for. One is to see into things that we can't otherwise see into, make things visible that are invisible. And the other is to look at very, very small structures, at molecular and atomic structure. Okay? And so the way that we've used X-rays, the technologies, we'll get to in a moment. And so just reinforcing about the spatial scales that we look at, if I grab a cursor here, you see this green arrow on your screen. You see at the millimeter scale, we can use visible light and all the way down to about 10 to the minus 6 meters or 1 micron. That's the size of a cell, roughly. We can resolve those with microscopes and with visible light. To go to even smaller length scales, down to viruses and molecules and individual atoms, down to the nanometer and tenth of a nanometer or 1 angstrom scale, the scale of materials, the fundamental physical structure of them, that's what we need X-rays for. And so far we've been talking only about images, about still images. We see the image of the hand or of the table salt, and it's not moving. There's no time dynamics involved with it. And what we're moving toward with our X-ray technology now is into very fast time scales. So we'll be talking a little bit about the different time scales of different physical processes, going from nanoseconds. So if you have a gigahertz processor in your computer, then it's doing calculations every nanosecond. And as you get down to picoseconds, that's the scale of nuclear motion of proteins folding and unfolding, actually are in the nanosecond to picosecond scale. And below 1 picosecond, that's the scale of chemistry, essentially, of ultrafast reactions of, say, light absorption in photosynthesis or in our vision. The first steps in vision happen at these extremely fast time scales, at femtosecond time scales. And those are the shortest pulses that we've been able to make with lasers. And just in the last few years, we've started to have X-ray lasers at the large facilities that reach these time scales. And so we're actually able to not just look at chemistry from the point of view of chemical reactions before and after, but to actually track those reactions through the process, to look at the exchange of electrons as chemical reactions occur in other very fast processes, energy transfers in atoms themselves. So that's where we're headed. And if we look back at how X-rays have been produced originally, I'm showing on the left-hand side here a tube from the early 20th century. And so this is the tubes that are basically still used in your doctor's office or in hospitals. Even advanced CT imaging is still done with this type of device. There was a bit of a revolution in the 1960s where they made a disc spin inside the tube, and that, this is the spinning disc, and that allowed them to up the power by a factor of 100 or so. But really, the X-ray flux and its brilliance, the two key aspects of these X-ray sources, hasn't changed much in 100 years. And so of the roughly 30 Nobel Prizes awarded for X-ray science, more than half were done with these small tubes, actually. But that was primarily before the late 20th century. And since then, a different type of X-ray source has taken over. So starting around the 1980s and then became much stronger through the 90s and even to today, these synchrotrons that were originally designed for high energy physics were optimized to produce intense X-rays. So these things are large. If I move my cursor, you can see these are cars in a parking lot. This is the Advanced Photon Source outside of Chicago, and there's about 50 beam lines. So there's 50 groups of scientists simultaneously taking data on this. Thousands of people per year go to this and get their X-ray data. And the science that they do is really quantitatively different than what can be done with those small tubes. The X-ray beam that comes out of this is between 1 billion and 1 trillion times brighter than the X-ray tubes. It's just, it's like if you were talking about computers, the X-ray tube is an abacus, and this is a modern microprocessor. That's the difference in the scale of science that you can do. It's really tremendous. So these are great, and most of the recent Nobel Prizes in X-ray sciences have been generated by science done at these devices. But as you see in the numbers here, that it's kind of a mile in size and $1 billion in cost. And so there aren't a lot of them around the world, and you're obviously not going to have that in your local hospital. And a further development, also using these big particle accelerators. So that's a synchrotron, which is a type of particle accelerator where there are electrons that go round and round inside that device producing X-rays. And starting in 2009, this device came online. This is the Stanford Linear Accelerator out in California near Stanford University. And this device is a long straight line, a giant particle accelerator. It's 2 miles long, and it zips the particles up to very high energy, and then they wiggle in a magnet and produce an X-ray laser beam that then proceeds down here and into experimental halls. This is a highway seen from above. This is Interstate 280 crossing over this large device. And so it's a remarkable thing. It's the world's first X-ray laser, but again, it's a $1 billion device and it's 2 miles long. What we're trying to do is shrink that to what you see in this upper picture, where this human figure is standing in front of an accelerator. Our accelerator is literally 1 meter long or 1 yard long. And then there are some magnets and things that manipulate the electrons. And then our electron beam collides with a very powerful laser pulse down here and produces X-rays so that the X-ray experiments can be done here. And so we shrink this 2 mile long device down to something it's about, our whole device actually is a little bigger than what's shown here. It's about 30 feet long. And it could be smaller, but this is the very first one, so it's a little bigger than it possibly needs to be. And so I want to talk a little bit about, how can we take a 2 mile long thing and make it into something that fits in a basement lab. And one of the tremendous differences is that for the large machines, they have an energetic electron beam that then goes into something called a wiggler magnet. And it wiggles the electron beam. If you picture an electron beam going along at the speed of light, and it goes in between these magnets, which look very much like refrigerator magnets. Each one is a couple of centimeters long. And the electrons, when they get in that magnetic field, they wiggle back and forth. And in doing that, they produce this X-ray laser light. And in order, there's a relationship between the electron beam energy and the period of the magnets. And because the magnet period is a couple of centimeters, the electron beam has to be really high energy. Thus, we need a 2 mile accelerator. If we can replace those magnets with a laser pulse, the laser field has an oscillating magnetic field, just like that wiggler magnet, but it's 10,000 times shorter period. Instead of centimeters, it's a micron. And what that means for our electron beam is we can shrink that 2 mile long accelerator down to 1 meter and produce the same X-rays out at the end. They're mostly the same, but they're not as powerful. So our device is about 1,000 times less powerful than the big ones. But this big one, I should have mentioned in the previous slide. I said that those big rings were about 1 billion to 1 trillion times brighter than a lab source. This device is another factor of 1 billion brighter than the rings. And so just the orders of magnitude are a little bit mind-boggling. And so even though we're 1,000 times less powerful, we're still up in the billions more powerful than other choices for a small lab.
Mike, I see there's a question from our audience. What wavelengths can this compact laser produce? How short, how long? So you're going to get into some of this, I know.
Sure, I can answer that quickly. It's tunable, which means we can change the X-ray wavelength or the photon energy. And we're looking at going from a few nanometers wavelength down to below 1 angstrom. So in our first version, we're powerful X-ray pulses, but not yet a laser, and that will actually go up to what we consider high photon energies around 35 keV or kilo electron volts. So that's a good medical X-ray energy, and that can go down in terms of X-ray energy down to about 500 electron volts. So very widely tunable by changing the electron beam energy. Okay, it looks like that answers the question. And the pulse lengths are interesting too in terms of short and long. Our typical pulse lengths will be about 300 femtoseconds in the initial version, and then with our upgrade to the X-ray laser, they'll be below 10 femtoseconds, and we believe they'll be below 1 femtosecond into the attosecond regime. And that's the time scale of an electron going around a hydrogen nucleus. And so it's really the fastest time scales we have in nature, in kind of ordinary material science. And so we should be able to track the very fastest processes. So this is smaller. It's 100 times, it's about 1,000 times smaller and about 100 times cheaper. So instead of $1 billion, it's a kind of a $10 million instrument. People usually want to know what it costs. So there's still a particle accelerator, there's powerful lasers, there's still complex equipment involved.
But it's the kind of price that you could have, and a hospital might be able to afford.
Yeah, it's not too different than a state-of-the-art MRI machine or CT setup. It's similar, or electron microscope. It's a similar scale. I wanted to say just a little bit. I'm located both in the physics department and in the Biodesign Institute at ASU. And so this Biodesign Institute is a very multidisciplinary organization. My background is as a particle accelerator designer. So I've worked on those large machines for much of my career, and now I'm shrinking it to design this small one. And I spend my days working with biologists and chemists and materials people, designing the X-ray beam that's best for their science. And so we're a very diverse group in the Biodesign Institute here at ASU. And they did build a new building to house this device, and so it's for me and 200 of my closest friends, I would say. And so we're building that. And now I'm going to go into a little bit of what we can do with this. So when we make these short pulses, you have to understand those old X-ray machines had a continuous beam that was on all the time until you switched it off. And the way ours works is it makes very, very short pulses of X-rays, intense pulses. And so we can track, what this is showing is a diatomic molecule, and the two atoms are vibrating as they do. And if you diffract X-rays from this two-atom system, what you see, let me use my cursor here. So this is the vibrating molecule, of course, the two atoms. And depending on how far apart they are, this pattern over here is changing. This is the diffraction pattern. So we would have our X-ray beam hitting the atom, scattering from it, and being recorded on a detector. And we would have these intensity variations in the X-rays. And if we keep taking many shots, it's really actually just how a real movie is made with a movie camera. It takes many still frames, and then you scroll them by. And we do the same thing with our X-ray devices, where we have a fast detector that can take 1,000 frames a second, and we can track these. Each one is short on the time scale of this vibration. Vibrational periods are typically around a picosecond or a few picoseconds for molecules in motion like that. And so we can not smear out this pattern, which you would get by integrating over a long time, but take these very fast snapshots and then make a movie with it.
So Bill, I guess what you're saying is you've slowed down this cartoon here for us to see, but the molecules are vibrating with a period of about a picosecond. I think that's what you said.
Yes, that's right.
But then your X-ray pulse goes through 100 or 1,000 times faster than that.
Right.
Femtoseconds.
Yeah, femtoseconds. Yes.
So the X-ray sees the molecule is stationary essentially as the X-ray goes through, and that's why you can get that still picture of its condition.
Yes, that's right. We take stroboscopic shots of it. Okay, and let's see, we have another question. Are these short pulses energetic enough to potentially cause damage to materials? The answer is yes for that. There are different ways of operating this. Some of our intensities are low enough that they don't damage the material or have minimal damage. The big devices, the X-ray laser out at Stanford has extremely powerful pulses, and they do actually destroy the atoms that they hit when they hit them. And so what we do is spray crystals across the X-ray beam, and so that each shot of X-rays that comes along hits a different crystal and then diffracts off it to make the pattern like what we see. And even though the crystal and the atoms are destroyed because they're highly ionized, they pull all the electrons off them, we get our diffraction pattern before that destruction happens. So that's over a time scale of tens of femtoseconds.
I think you had a name for, isn't there a name for that technique?
It's called, diffract before destroy.
Diffract before destroy, yes.
Yeah, okay. Type thing, or the fancy name is serial femtosecond crystallography. And so you spray the atoms across the beam in jets, and that's what this picture on the left is depicting, that each shot is going to hit a different vibrating atom. And then we have advanced software algorithms to reconstruct from all these different atoms the vibrational periods. Okay, and we do also intend to use these for medical imaging, and of course there where you have a patient, you're not looking to destroy tissues. And so they don't have to then be a trillion times more powerful. They have to be what tissue can take, living tissue. And I'll get to the type of imaging that we do. It's actually more sensitive than conventional imaging, and so it has less radiation dose. Okay, so let's run. I've talked so far about how we make X-ray pulses and how we're going to shrink the big device into a small device. And now let's talk about the science that's done. So in one of those initial images I showed was of table salt, which was a very simple atomic structure, just the two atoms in there. And now these are atomic structures. These are proteins, very complex protein complexes that have to do with biology. And so, grab my green cursor here. So on the upper left, we're seeing what's called photosystem 1. This is what's in plants for photosynthesis. It's 99.5% efficient in converting light photons into energy, electrical energy.
So Bill, this is part of the chlorophyll molecule, is that correct?
Yes, that's right. That's right. And so it has a very complex structure. We know what the static structure looks like. And what we're trying to do now is to look at the energy transfer that happens on these femtosecond timescales as it absorbs a photon and transfers that energy through a set of complex reactions into an electrical signal. And so this is the much more complex structures that we can look at now that we have these very powerful X-ray beams. And now the X-ray lasers have given us the ability to look at the time dynamics, so getting not only the structure but the dynamics too. And the lysozyme enzyme is another protein that's well understood, and we often use this to characterize our X-ray sources and make sure that we are correctly interpreting the data from everything from the sample delivery to the X-ray beam to the detectors and so forth.
And did you say before this is what, egg whites?
Yeah, this is material for egg white. Okay, so that's diffraction studies, let us see that, and some of the uses of that are in drug discovery. So in making new pharmaceuticals, particularly understanding side effects and what can potentially block receptors. Often drugs are targeted at particular receptors to block a certain type of immune reaction. And so the pharmaceuticals have to screen many candidates looking at different drugs and how they interact. And going from just the static structure to actually looking at the function and how they bind is key to having a much less time-consuming pharmaceutical development. So Kirk Clark, head of X-ray scattering at Novartis, is very interested in having such a small source actually in the basement of his own labs. I'll talk a little bit about medical imaging. We saw the hand with the ring early on, and using conventional X-ray tubes, a modern one, if we were looking at a cancerous liver, we would see an image like this. I'm sorry, let me use the, on the left side, an image like this, where this red line is actually surrounding the cancer tumor. And obviously it's not visible at all. That's because it's the same density. Basically all soft tissue is roughly the density of water, and so does not absorb X-rays in a differential fashion. When we have coherent X-rays, then we cause shifts in the phase of the X-rays. The index of refraction, the phase part of that is about 1,000 times more sensitive than the absorption part. And so we can detect these small changes in phase and see lots of the granular structure in individual soft tissues. Easily makes out the difference between the healthy tissue and the tumor. And so, and it does this at lower dose. So we're very excited that we believe we can really revolutionize medical imaging with this, and we're working with the Mayo Clinic on that effort.
So a question from the audience. Bill, this is a sample that has been taken from someone's liver, I imagine.
That's correct. Yes.
Now, there's a question, could we image a live cell at nanometer resolution? Because, you know, clearly that might be a holy grail, but I think a live cell might be challenging.
Yeah, you can ask what's the resolution of this, and that resolution is typically set by the field of view, and we're looking at medical tissues, or human tissues or animal tissues. You're usually looking at centimeter scales, and so we can resolve. It depends on the detector you have. If the detector is, say, 10 megapixels, an excellent detector, then what does that go down to? That would go down to roughly 1/1000th of your field of view. So you get into the micron range with a centimeter field of view. The X-rays don't limit the resolution. With a particular type of imaging, microscopy, called coherent diffractive imaging, we can get down into the nanometer range, down to about 10 nanometer resolution in real space imaging, not diffraction.
Sounds like you have plenty of image resolution. I guess this is a high vacuum system, essentially, isn't it? So it would be hard to put a living cell in there.
No, it doesn't have to be in vacuum. This can be under biological conditions, so wet, and at room temperature. So high energy X-rays, which are what are used for medical imaging, tens of keV, easily propagate through air. Yeah, for some of the material studies with soft X-rays, they don't propagate through air; they're absorbed. Then we have to do it in vacuum. But the medical imaging does not need to be in vacuum.
Thank you.
Sure. Okay, here's another image. This is showing a cross-section of a wrist from a cadaver, actually. And so here's the MRI image. You're seeing cross-sections of bone in these dark areas. And then if we look at the labels, we can see there are tendons and nerves and blood vessels and different parts of the flesh here are visible in the MRI, but in phase contrast has much higher resolution. You can see that there's both better contrast and a higher resolution. You can see the interior bone structure and make out lots of the different parts of the flesh, from the skin and into the arteries and capillaries and so forth. So really tremendous, can have tremendous impact on medical imaging. You can see plaques inside of arteries, lots of different things that currently are invisible.
So a lot more structure visible in the image. Somebody had a question, does coherence length of the laser matter? What is the coherence length?
Coherence length is the distance over which your wavefronts are in phase. if that's helpful. And yes, the coherence length, a large coherence length helps you. You basically like your coherence length to be the size of the beam itself. And I didn't go into it, but our source size, where the fundamental source of the X-rays here is the collision point of the electrons and the conventional laser that then converts into X-rays. And that's only a couple of microns in size. That we focus both the electron beam and the laser beam down to a few microns. And so that's our source size, and we would like our coherence length to be that. We, in our initial devices, actually not yet an X-ray laser. It's partially coherent, and it can do this kind of phase contrast image. This phase contrast image was done at a synchrotron, a partially coherent source, not an X-ray laser.
Interesting. So you don't need full coherence to see these images?
No, you need partial coherence. You need better coherence than you get from the tubes, but you don't need the full coherence of a laser. And the laser can be helpful, but lasers are... we're all familiar with laser pointers. They're extremely useful for what they do, but you wouldn't use it to light up a football stadium. And so there are times when a laser is the right tool, if you're looking at intense things and things that respond to high coherence, and there are times when you need more of a brightly lit area to look at these large field of view type things. So our laser spot size tends to be at the microns scale. You would not see, centimeters like this. Okay, let's keep going. I'll go a little bit quickly now. This is looking at carbonate reservoirs. If some of the oil-producing and gas-producing countries are very interested, if they can squeeze more, a higher efficiency of oil out of the rock, then it has large economic impact. And so really understanding the porous structure of the oil-bearing rocks is very useful. And so Maersk Oil is interested in being able to do this in the field. This is a fun one, cultural studies. This is at the Louvre. We were invited over there a couple of years ago. They have 200 PhDs in the basement. They don't bring those artworks to a synchrotron. They can't do that. They stay in the museum. So on Mondays when the museum's closed, they bring them downstairs, and they have a wide range of analytical equipment down there. They have older X-ray sources. They're very interested in having one of these small particle accelerators in the basements to do artistic studies or study the materials and the methods and what might lie underneath. And I think I have, this is a favorite description. And so this is a painting by Edgar Degas that hangs in Australia. And if you look at the person's face, you can see that there are some dark areas where another painting is starting to show through. That's not her painting, but the paint is starting to fade, and there's clearly another painting underneath, and they wanted to see if they could discover what it is. And so they did bring this one to a synchrotron, put it in the beam, and we're able, if I morph now away from that painting, able to see the outline of a different woman, a different model. This is a well-known model of Degas. Her name is Emma Dubigny. Could see the outline of that just based on the density that's underneath. Could kind of strip off without damaging the painting that. And then furthermore, using X-ray spectroscopy, could determine the different pigments that are present in that painting. And so by scanning the X-ray energies, are able to determine that and actually able to recover the original colors of it. And so this is all without disturbing that surface painting, are able to fully pull out the painting that's underneath. Really quite a remarkable study.
Isn't that remarkable?
Yeah,
look at that.
Yeah. This was done in Australia at the Australian synchrotron. And so to be able to do that right within the museums is very interesting. Okay, moving on. Okay, so far I've described a lot of these applications actually can be done with these short and intense X-ray pulses like a synchrotron. What we want to do is truly revolutionize the world and make an X-ray laser out of it. And to do that, I'm showing in these two images all these little kind of BBs represent the electrons. And when we make an electron bunch, it's a very short bunch, femtoseconds long, but the electrons are not arranged within that bunch. They're just shot noise. They're randomly spaced at the X-ray wavelength scale. That's what lambda x is. And so they all start wiggling in a magnet or in my laser beam, and they wiggle out of phase. There's no phase relationship. And that's what gives us very intense X-rays, but not fully coherent X-rays. To be fully coherent, we need to do what's depicted in the bottom picture, which is to get the electrons to arrange themselves in little bunches separated by the X-ray wavelength. Keep in mind these X-ray wavelengths are about 1 angstrom, an angstrom to a nanometer. And so we're looking to have structure in our electron beam at the angstrom to nanometer scale and freeze that in. There's a natural process that happens for the big machines where they can start up from the random distribution, and then the X-ray radiation acting back on the beam generates this after after 100 meters, and an undulator can do that. That won't work for our small machines. So what we plan to do is arrange the electrons before they start emitting X-rays, arrange them into these small packets. And in order to do that, we have fabricated these nanoscale gratings. So this is a 200 nanometer thick silicon single crystal membrane. These particular ones are supplied by Norcata, and Trevor is working on some for us now in-house, where we have a 400 nanometer period of silicon spaced by vacuum. And by scattering an unpatterned electron beam through this grating, we will get a density variation across the beam. And this is actually an electron micrograph where the bright stripes are where we have electrons and the dark areas are where there are no electrons. And that is just the kind of pattern that we need for our X-ray laser, but we have to shrink it down now from a few hundred nanometers down to a nanometer. And we have to swap it from the spatial scale into the time domain, into the temporal scale. And I won't go into the accelerator physics of that swap. Let's just say it's a little bit exotic. But the shrinking of this pattern down to the nanometer scale is not difficult. That means we have demagnifying optics of about a factor of 100. Electron microscope has a magnification of 100,000 or more. And so demagnifying by a factor of 100 is relatively trivial from an optical perspective.
So it's a magnetic lens of some form? It's a lens, yeah.
Yes,
okay.
A series of magnetic lenses demagnify. And then we are able to swap it from the space domain into the time domain using an accelerator physics trick called emittance exchange. And so this is, again, showing that density pattern in the transverse space. And so if we project that onto the x-axis, we see that we have a rapid variation in the density, here. And that's exactly what we need to make an X-ray laser. We just need to now swap this into the time domain. And so FEL means free electron laser. We have something we call the bunching factor. And this is what we want is for this to be, have some magnitude above 0, and this is a beautiful beam. This is a nearly perfect beam to laze with.
So you talk about being monochromatic, right? It has a single wavelength. Is that the goal?
At times, this is showing, if you have a grating with a single period in it, then you'll get a single wavelength out. This is looking at essentially the Fourier transform of that. So you see the fundamental, and then it has a couple of harmonics present, which is fine. So we would choose to laze on the fundamental. But we can also do, we could have a chirped period, and basically anything you can think to lithographically pattern into the silicon, we can then imprint on the electron beam, which then transfers that to the X-ray phase fronts. So we can have multiple colors. We have different colors separated by attosecond to femtosecond time delays. We can have actually phase locking of the different colors because it's deterministically produced on every shot. So we think this will tremendously advance our ability to control X-ray beams. And so this chart probably doesn't mean much to non-experts, but we are plotting here peak brilliance in what I'll just call standard units, and this is a logarithmic plot, so each one's an order of magnitude, versus photon energy. So 1 keV is right where my cursor is, and 10 keV, these are kind of the scientific workhorses, and then up to 100 keV is where you might do medical x-rays. This is showing lasers are at low photon energy, but extremely bright. These are conventional lasers. And then synchrotrons, these are our very bright synchrotrons, 1 billion times brighter than your lab source now. The XFELs are 1 billion times brighter than them and in the X-ray wavelength range. We're not quite as powerful as those big XFELs, but we're much brighter and at shorter wavelength than any competing technology. So we're very excited about where we're going with this. And I think that wraps it up. So with that, I'll take any questions or have some discussion.
Well, thank you, Bill. The thing I find so exciting and fascinating about this is the way it combines these multiple scales, right? You have the dimensions at the sub-nanometer, you have times in the femtoseconds, you're building a machine that's relatively small, but it's still 20 or 30 feet, and yet it depends on a silicon grating that someone's made with 100 nanometer dimensions. And then the laser itself is a high energy, fast laser system, isn't it?
The colliding laser, yes,
Colliding
a very
laser.
Powerful laser, 200 millijoule picosecond pulses for that.
So it's this sort of multi-scale physics lab that is in the basement of this building. And one of the questions was the radiation safety issues. I mean, I know a lot of the design of the building was to operate the system safely, right?
That's right. It's a small particle accelerator. It's still a particle accelerator. There's significant ionizing radiation. The electron beam energy is about 35 mega electron volts, 35 MeV. And so when that hits something like a concrete wall or a metal beam pipe, it makes gamma rays. And so we have thick walls. Their walls are 4 feet thick in the laboratory. Around the accelerator itself. It doesn't need to be that thick around the X-ray experiments, but just around the accelerator it has to be thick wall.
So I was lucky enough to get a tour a few weeks ago, and yes, big concrete walls, huge steel doors that slide into place when people are not meant to be in the room, and all kinds of facilities. So yeah, a state-of-the-art lab for a state-of-the-art multi-scale physics facility. So I'm looking at the list of questions. I don't see any new ones. So Mike, maybe we hand back to you.
Yes, thank you, Trevor. I do have a question, maybe a comment more than a question, Trevor. You know, it's sort of funny. Just a few years ago, this was like a dream, right? There was these huge, synchrotron sources to do this sort of work, and everyone sort of imagined what would it be like if you could actually do this in research lab scale, and here it is. It's almost there. Bill, you mentioned it's not exactly a laser yet. What is your time frame for when that might happen?
Well, we're in the assembly phase now. On the first phase, we'll be turning that on in the coming months over the summer, and by the end of the year, producing our X-rays, and then. We seek additional funding, I'll say, to complete the laser version. The laser version is not a huge upgrade of the existing one, but it takes some more equipment, and so we're talking to the federal agencies about funding for that. So it depends on that time scale. Once the funding is obtained, it would take 2 years to implement, and then assuming it doesn't work on day one and there's some tuning and learning going on, it would take another couple of years after that. So it could be a kind of a 5-year time scale. There's
another question, Trevor and Bill, that you see came in from our colleague at the University of New Mexico. He was aware some time ago of another approach to making these devices using a standard. I'm not sure exactly what the deal is through a crystal. Do you see his comment there? Are you aware of anything like that?
I do see that, and I think I know what that refers to. Let's see, X-ray. So people have worked on having an electron beam go through crystals, and we've talked about wiggling the electrons, right? The big FELs wiggle the electrons with these refrigerator magnets. My concept wiggles them with a laser beam, and that wiggling, so the refrigerator magnets wiggle them with centimeter period, we wiggle them with micron period. A similar effect happens when an electron channels through a crystal along a crystal axis. Through the atomic potentials, it can also wiggle and make something called channeling radiation. And that's at an angstrom scale, so very low energy electrons, non-relativistic, can actually make X-rays doing that. I believe that people have observed that channeling radiation as a physics experiment, but there's been quite a lot of effort to make that a practical device, and it's tough. I would say. I think the photon count is quite low.
Doesn't sound like it would be a very bright source.
No, I think not. I think it's interesting that the physics works, but it's not useful to do science. So the type of laser collision we use, that process is called Thomson scattering or inverse Compton scattering. And other X-ray, it's not a new idea. That's been known for many decades, that physics, and people have done the experiments. But nobody has yet built a useful X-ray source on it. Actually, I should mention there is one. There's a small synchrotron at TU Munich that does that. So it's a ring-type device, not a laser. But using that laser backscattering, it's a whole nother ball game to just demonstrate the physics of the X-ray production versus making a beam that's useful for the biologists and the chemists.
Dr. Graves, I actually see the unique aspect of your work is this collaboration. You're headquartered right there in the Biodesign facility, and it's not that it's forcing it in any way, but it's by design, I guess. I don't mean to misuse the word, that your physics work is collaborating with the folks working on biological systems. Pretty unique.
Yes, that's right. It's a change of pace for me. I've been embedded in physics labs much of my career. I'm 60. I'm not young, and it's really been just absolutely fascinating working closely with the biochemists and the materials folks and so forth. And for them, when an X-ray user goes to a beam line and gets their beam time and does their experiment, they're really just a customer with very little control over the source properties, over the X-ray beam properties. But here we're completely integrated together into this diverse team. They have a lot of input into the design of the device and what the beam is capable of doing, tuning energy or varying polarization, all these different interesting experiments. And so it's a much more collaborative effort between the users of the X-rays and the designers of the X-ray source.
And then what? Yeah, go ahead, Trevor. Yeah.
There's one more question. Your device could be used for Liga-type fabrication. That's a well-known microfabrication molding technique. But
the semiconductor folks are real interested in this thing, but not for fabrication, because they would need kilowatts of average power to have the throughput that they seek. And we're not in the kilowatt range, to put it mildly. But what we can do is metrology. So we can look at masks and lithography. They're operating, I think you call it the 7 nanometer node now.
That's right.
And they can't see it. The conventional ellipsometry measurements don't work. And so to embed these into the fab plant, the X-ray device, so that you could do small angle scattering and actually see in real time as the wafers go by the structures, is one of the potential applications.
Trevor, it just occurs to me that some of the most interesting things always happen at the crossing of these fields. Isn't that the case here as well?
Very true, isn't it? It's going to be exciting to see once the beam comes online, and then all the exciting science discoveries that it will enable. And then who knows, maybe they'll be in every hospital, just like we have MRI machines in a lot of hospitals.
Yeah, our goal is to get it out. It's people ask, can they come use it? And I say yes, but you can have one yourself too. You don't have to come here. to do
Well,
it.
colleagues, that brings us right to the end of our presentation today. And Professor Graves, thank you very much. Just an excellent presentation. Trevor, thank you for being the color commentator there, working with Bill side by side. Colleagues, I'm going to now ask you just to answer some brief questions on a survey. And here's what's going to happen. I'm going to click on the survey link. And presenters, please don't click on this link. A new browser window will open, and you'll be able to do the questions. There's one or two machines this may not work for, so here we go. We're still live with the audience. Again, Professor Graves, thanks very much for your presentation today, and Trevor Thornton, thank you for helping in the production of this webinar series. Colleagues, that officially ends our webinar today. This webinar has been recorded. I'm going to turn off the recording now. You'll automatically get a link to the recording. It usually takes us a few business days to process that. So thank you and goodbye, everyone.